Hi everyone, welcome back. Today we will be discussing the acid-base chemistry of amino acids. As a general overview, I will answer the following questions in this video. If you want to skip to any specific part, you can do so by clicking the timestamps found in the description section below. I will give a brief rundown of amino acids and how they relate to acid-base chemistry, why amino acids are considered amphoteric, what is an acid and what is a base, what is pH, and what is pKa, what happens to an amino acid when it's in an acidic condition, what happens to an amino acid when it's in a basic condition, and what happens to an amino acid in an intermediate pH condition. Let's get started. A brief rundown of amino acids and how they relate to acid-base chemistry. Amino acids are building blocks that create proteins. All amino acids follow a basic structure. They contain a central carbon atom. This is often called the alpha carbon, an amino group, a carboxylic group, a hydrogen atom, and a side chain. The side chain is often referred to as the R group as well. The reason why amino acids are discussed in acid-base chemistry is because they contain the amino group and the carboxylic functional group. The amino group is basic or alkaline in nature, and the carboxylic group is acidic in nature. This is why amino acids are also considered amphoteric. By definition, an amphoteric molecule is a molecule or an ion that can act both as an acid and a base. Because amino acids contain the carboxylic acid group and the amino group, they behave both as an acid and a base. So what are acids and what are bases? Now, acids and bases can be defined in many ways. The three main definitions that come up in the MCAT and almost all undergraduate biochemistry classes are as follows. So we have firstly the Arrhenius definition, the Bronsted-Lowry definition, and then the Lewis definition. In the Arrhenius definition, acids are molecules that donate a hydrogen ion. This is also called a proton when acids are dissociated in water. Bases, on the other hand, are molecules that donate a hydroxyl ion, as in an OH group, when bases are dissociated in water. Now, the second type of definition, which is the Bronsted-Lowry definition, states that an acid is a molecule that donates a proton, which is a hydrogen ion. So these are proton donors. On the other hand, a base is a molecule that accepts a proton, that is, again, a hydrogen ion. So bases in the Bronsted-Lowry definition are proton acceptors. Thirdly, we have the Lewis definition. In the Lewis definition, acids are molecules that accept a pair of electrons, and bases are molecules that donate a pair of electrons. All these definitions can be confusing. However, a good thing to remember is that you don't need to know all of these to understand the acid-base chemistry when it comes to amino acids. Each of these definitions comes in handy when discussing specific topics in chemistry. For our current purpose, the main focus would be on the Bronsted-Lowry definition. So moving forward, remember that we are working within the framework where acids are considered as molecules that donate a proton and bases are considered as molecules that accept a proton. Now that we have a definition of acids and bases, let's go to the next topic, which is what is pH? At the simplest level, pH is a quantitative measure of a solution's acidity or basicity. We learned that acids release protons and bases take on protons. Therefore, a solution's acidity and basicity depends on the amount of protons found in the solution. Now, you might wonder, okay, that's not difficult to grasp. Why can't we just count the amount of protons and classify a solution as acids or bases depending on that? And you are correct. You can do that. But when we're dealing with ions that are so tiny, and are present in such large quantities, the amount would be impractical to actually count and discuss in terms of real numbers. So scientists created an easier measure, which is the pH. Simply put, pH is equal to the minus log of the concentration of protons. Therefore, pH is a simple calculation where you get the negative log of the concentration of protons in a solution. P is used to represent minus log, and H plus within square brackets is used to represent the concentration of protons, or again, proton meaning a hydrogen ion. Let's understand this by using an example. Consider this question. What is the pH of 0.0235 molarity of hydrochloric solution? Now, if you recall your chemistry, you know that hydrochloric acid, which is HCl, is a strong acid and therefore will dissociate completely. So in this case, 0.0235 molarity of hydrochloric acid would give you equal amounts of H plus and Cl minus, meaning the concentration of H plus that we would get is 0.0235 molarity. So all you have to do to find the pH of this specific solution is to input 0.0235 molarity of protons into the pH equation. The pH in this circumstance would be equal to negative log of 0.0235. This can also be written as pH equals negative log of 2.3 into 10 to the power minus 2. This is just the initial concentration represented in standard notation. Writing the equation in the second way that I mentioned gives you an easy way to ballpark what the pH of a given solution is, especially for the MCAT when you don't have a calculator. Having an estimate or a good way to ballpark an amount would be important and would save you a lot of time. So you write it in the standard notation and your pH would be around the value of whatever the 10th to the power value it is. So in this case, the pH would be around 2, meaning when you write an amount in the standard notation and you always get a 10 to the power value, that is going to be the value at the top is going to be closer to what the pH would be. If you add it to your calculator, you will find that the accurate value is 1.629, which is very close to 2. Again, for the MCAT, estimations are good enough. You don't have a calculator and they don't expect you to make these time-consuming calculations. And the answers that they usually give as response options are quite far apart, where having a ballpark or an estimate would be sufficient to pick the right answer. Now you might be wondering why these chemistry calculations matter when we talk about the acid-base chemistry of amino acids. I think they're important because it's really useful to understand the foundational concepts underpinning these topics, so you don't have to end up memorizing certain things and you can truly understand why an answer is the way it is or how to derive at an answer. So let's just keep going. So now we know how to calculate the pH, but a number means nothing if we don't know how to interpret it. So this is when the pH scale comes into play. The pH scale ranges from 0 to 14. Roughly speaking, if a solution is in between the pH ranges of 0 to 6, it is considered acidic. If it is between 6 to 8, hovering around 7, it is considered neutral. And if the solution is between 8 to 14, it is considered basic, or in other words, it is considered alkaline. So the lower the pH, the more acidic a solution is, and the higher the pH, the more basic or alkaline a solution is. This is a little counterintuitive, and you might be tempted to think that because we are using the concentration of H plus ions, also known as protons, in a calculation, the higher the pH, it would likely represent high H plus concentrations, meaning it would be more acidic, but this is not the case. You need to remember that we are taking the negative log. So the pH and proton concentration are actually inversely related and not directly related. Now, we have almost all the information we need about acidity, basicity, and pH to understand the acid-base chemistry of amino acids. However, we have one more thing to cover. That is the pKa. What is the pKa? Remember, when we were talking about pH, I mentioned that P is used to represent a negative log. So in pKa, it represents the negative log as well. It just represents the negative log of a different thing. In this case, the negative log of Ka. But now we have a new term, Ka. But what is Ka? Ka is the symbol we use to represent the acid dissociation constant. Acid dissociation constants are used to quantify the strength of an acid. Again, the main point here is the strength of an acid. pH is used to classify if a solution is acidic, neutral, or basic. Ka is used to represent how strong an acid is. Acid dissociation constants are intrinsic characteristics of molecules. So scientists have worked really hard to identify these constants. You will see this equation when you're studying acid dissociation constants. This indicates how much proton ions will be released by an acid when it dissociates in a solution. If more H plus or proton ions are released, the acid is strong. If less is released, then it is considered weak. Therefore, Ka is directly proportional to the proton ion concentration. Therefore, Ka is a representation of how strong an acid is. Now recall that previously I said why we use the negative log of something is to make things more comparable. So the same concept applies when it comes to acid dissociation constants as well. So however way you have the value of Ka, and usually these values are given to you within the context of a test. You can plug it into the pKa equation in order to get a pKa value that helps you to easily compare the acid dissociation constant of one acid to the acid dissociation constant of another acid. So if an acid releases more proton ions, it's going to have a very high Ka and a lower pKa. You are not required to know the Ka or pKa values of molecules for the MCAT. If a question relates to one of these, you will be given these values or you will be tested on information that does not require you to recall. Okay, why does all of this matter? What does pKa have to do with acid-base chemistry of amino acids? This matters because amino acids have two functional groups with acidic and basic properties. The carboxylic acid group is acidic and the amino group is basic. The carboxylic acid can donate a proton and the amino group can accept a proton. Therefore, all amino acids have at least two pKa values. The pKa of carboxylic acid groups is around 2, and the pKa of amino groups is around 9 to 10. Some amino acids have ionizable side chains or R groups. So these could also contain acidic or basic groups, and therefore these themselves could have their own pKas indicating how strong their acidic or basic group is, or weak that group is. These pKa values help us to determine how each amino acid will behave in different pH environments. And this is the entire goal of this video. A lot of questions on the MCAT will test you on how a specific amino acid will behave in a specific pH environment. Please note that pKa values you see will be slightly different based on the source you look at, because these values are dependent on environmental factors like temperature. So if the conditions aren't standard, this is going to vary. But they usually hover around the same values when it comes to test questions that you're going to be given, because most of them are likely under standard conditions. In addition to these values, you will also need to know one key point to understand how amino acids will behave in different pH environments. Ionizable groups gain a proton in acidic environments if the pH of that environment is lower than the pKa of that ionizable group. Similarly, ionizable groups will lose the proton in basic environments if the pH of that environment is higher than the pKa of that ionizable group. That is, when the pH of the environment is low, that is, when the environment is acidic, ionizable groups will be protonated. When the pH of the environment is high, that is when it's basic or alkaline, ionizable groups will be deprotonated. Let's think about it like this. Let's say that you have glycine, which is an amino acid, at 3 different pH conditions. Recall that glycine is the simplest amino acid that there is and has a hydrogen atom as the side chain. So the only 2 ionizable groups in glycine is the carboxylate group and the amino group. Let's first consider the acidic condition. Okay, so we have our glycine amino acids in an acidic environment. So let's conceptualize what an acidic environment is. An acidic environment has lots of free-flowing protons, that is H+ ions. Let's say that the pH of this given environment is 1, extremely low, which means that it has extremely high amounts of free-flowing protons. The moment glycine molecules are introduced to this environment, its amino and carboxylate groups are going to get protonated. Just think about it like this. Think about having vacancies in the amino group and the carboxylate group that can only be filled by protons, and there are so many protons around, so they're just going to pop into those vacancies. That is a conceptual way to think about it, but when it comes to exam questions, unfortunately, you need to think about these in terms of numbers that are given to you, because it's not going to be that easy to figure it out. So let's consider the pKa values in relation to the pH of the environment, because that's how you will be able to figure out how a specific amino acid will react or behave in a specific pH environment. So the pH of the environment is 1. We decided that. The pK of the amino group, we know, is 9.6. So it was between 9 and 10, as we discussed earlier. And the pKa of the carboxylate group is 2.3. Recall that we discussed this before. Ionizable groups gain protons in acidic conditions if the pH of that environment is lower than the pKa of the ionizable group. Similarly, ionizable groups lose a proton in basic environments if the pH of the environment is higher than the pKa of the ionizable group. So in this case, The pH of the environment is much lower than the pKa of the amino group. Therefore, the amino group will be protonated. Because the amino group has a lone pair on the nitrogen atom, this lone pair will be used to form an additional bond with a free-flowing proton. This makes the initial neutral amino group now positively charged. More specifically, it's going to have a +1 charge. Now let's consider the carboxyl group. The pH of the environment, again, remember, is 1, is much lower than the pKa of the carboxyl group, which is 2.3. Therefore, the carboxyl group will also be protonated in this case. However, unlike the amino group, this will leave the overall charge of the carboxyl group neutral, because when a carboxyl group does not have the H plus or the proton, it's going to have a negative charge. So now that the carboxyl group has a proton, it's going to cancel out, and the overall charge of it is going to be 0. So in this case, glycine would have a +1 charge. So let's do the same exercise in a neutral condition. Let's say that the pH of the environment is now 7.4. In this environment, there is moderate amounts of free-flowing protons, not abundant, not scarce. The moment glycine molecules are introduced into this environment with a pH of 7.4, its amino groups are going to get protonated, but the carboxyl groups will not. In fact, the carboxyl groups will get deprotonated, making them negatively charged. Now, why is that? Let's consider this again in terms of numbers like we did previously. The pH of the environment is 7.4. The pKa of amino groups is 9.6. The pKa of carboxyl groups is 2.3. The latter two numbers were discussed earlier. They're not going to change. They're going to remain the same. Remember, again, ionizable groups gain a proton in acidic environments if the pH of the environment is lower than the pKa of that ionizable group. Similarly, ionizable groups will lose a proton in basic environments if the pH of the environment is higher than the pKa of that ionizable group. In this case, the pH of the environment is much lower than the pKa of the amino group. Therefore, the amino group will be protonated, making it have a charge of +1, much like in the first scenario. Now let's consider the carboxyl group. The pH of the environment is now higher than the pKa of the carboxyl group. The pH of the environment is 7.4, and the pKa of the carboxyl group is 2.3. Therefore, the carboxyl group will now be deprotonated under neutral conditions, leaving the net charge of the carboxyl group to be -1. Because of this, the glycine amino acid will now have an overall charge of 0, making it a neutral molecule in the neutral pH condition of 7.4. Why was it 0? Because the amino group got a proton, making the overall charge of that section of the amino acid +1, whereas the carboxyl group lost a proton, making the overall charge of that section of the amino acid -1. So +1 plus -1 is 0. Now, we've covered how glycine would behave in an acidic condition and a neutral condition. And now let's consider how it behaves in basic conditions. Let's say that the pH of the environment is now 11. In this environment, there is very minimal amounts of free-flowing protons. The moment the glycine molecules are introduced to the environment, the amino and the carboxyl groups of the glycine molecule are now going to get deprotonated. Let's again consider why this is using numbers. Just an overview, the pH of the environment is now 11. The pKa of the amino group is 9.6 as discussed before. The pKa of the carboxyl group is 2.3 as discussed before. Now, let's recall the rule of thumb, the statement that we all need to remember. Ionizable groups gain a proton in acidic conditions if the pH of the environment is lower than the pKa of that ionizable group. Similarly, ionizable groups will lose a proton in basic environments if the pH of that environment is higher than the pKa of an ionizable group. So in this case, the pH of the environment is higher than the pKa of the amino group. Therefore, the amino group will be deprotonated, making it have a net charge of 0. This means that it's just going to be neutral, unlike the case in the acidic situation or in the neutral pH environment. When it comes to carboxyl groups, the pH of the environment is now much higher than the pKa of the carboxyl group. That is, the pH of the environment is 11 and the pKa of the carboxyl group is 2.3. Therefore, the carboxyl group will be deprotonated in the basic pH condition, leaving the net charge of the carboxyl group -1. So now we have a situation where the overall charge of the amino group is 0 and the overall charge of the carboxyl group is -1. So the entirety of the overall charge of the glycine molecule when it's introduced to an environment with a pH of 11 is going to be -1. So that's it for this video. I hope this was helpful for you. If it was, please give it a like and please subscribe to the channel. It helps out a lot. And until next video, I hope you have a great rest of the day.